Forgive about that. In terms of housekeeping information, as Manya actually gently reminded me here, is that we are gonna have a break for lunch after my lecture, so we should be finished around 1, and between 1 and 2 we are gonna have a lunch break. During lunch break, if you're interested, you can participate to a tour of Aquabiotech facilities. So if you can actually tell us now, well, many people would be interested to go downstairs and have a tour at the RAS facilities. 1, 2, 3, 4, 5.
And people touring the facilities can still have lunch after?
Yes, because the tour is kind of going to be 15 minutes. So since lunch is going to be, as I said, for an hour, then you're going to have still time to have a networking lunch, interact, and then something to eat. So again, if you can please raise your hands. Okay, so we should have 11 people. Is it 2 groups? So I think that 11 people. I think that we 11 people. Usually we manage to bring one more. Okay. So during lunch, yes, and the bathroom is actually Manya just remind me. You get out of the room, turn left again, and then you find them at the end of the corridor. Now, during your visit downstairs, you're going to be accompanied by Dr. Simon De Guara, who's going to come and pick you up here because we're going to have lunch in this room. So that then you can go downstairs, have a tour of the facilities, come back up here, finish to have lunch, and then we're going to start with the session in the lab. So that's for, I think in terms of housekeeping, we covered everything.
Yes, just no phones downstairs as well. That's
okay.
Thing.
Yes. Please no phones downstairs.
And shall we tell them about tomorrow, about that option?
Yeah, we can address that either later on or so I can check if Brian is really managed to make it.
Okay.
He's in transit. So we can have the update later on.
Okay.
So thank you for your patience and now
give you the camera. So if you could use this at the back and take for MCST too. So good morning again, or better still, good afternoon. You have received quite a number of emails in the past 24 hours from us, actually in the past few hours, for reasons beyond our control, So Brian Hayden left is basically based in New Brunswick, so he had to take 3 or 4 planes to come here, and there were so many flight cancellations. Actually, at one point we were considering canceling this today's session, but since some of you actually took leave from work and so on, we said, listen, we still go ahead, and we're very grateful that you're still here, despite the many changes in plans and so on, okay? So what me and Alexia were discussing as well is that there is the option. I know most of you won't be available, but I'm just putting on the table that tomorrow, since Brian is probably arriving late this afternoon, okay, that tomorrow, for those of you who are available, we can still have a 2-hour lecture with him, okay, here. I know most of you took leave for today, but there is that option on the table as well. So probably during lunch, we will approach you to see how many of you are available for tomorrow and to pick the right time. Brian will be here throughout the day tomorrow, so if we can basically nitpick the right 2-hour slot. Even if there's 3, 4, 5 of you, we can do that session tomorrow as well. So at least, we have that exposure to Brian's expertise, So basically, I'll be speaking about the applications, a particular application of stable isotope analysis, which is relevant for Malta. As you might imagine, this is a relatively new field for Malta. Okay, so there haven't been many studies involving this technique for Malta. So that is why all of this interest. May I also stress that this is a project financed by MCST, by the Malta Council for Science and Technology, basically through its internationalization program. So this is a summary of what the general strategy, what the general plan is in terms of what we have in mind, how to use, how to apply for marine ecology stable isotope analysis, at least for the first few months. So we have 3 main targets. One of them is to evaluate the putative trophic impact, and here I stress, I'm being cautious using the term putative, okay, since obviously we are suspecting that there is a trophic impact of these invasive species because they are invasive and therefore they reach large numbers over a relatively short period of time, but obviously don't know as yet, since very few studies on trophic impacts of alien species have been conducted so far, not just in Malta, I'm saying across the Med and across the world in general. So normally when studies on non-indigenous species or alien species, because there's many synonyms, are conducted, it's basically impacts, the obvious impacts which are assessed, increases in population, perhaps displacement of the native species, but as such, trophic impacts on the food web in general are rarely assessed, even because of the inherent difficulties involved in assessing such impacts, okay? So we have, of these three targets, two of them, the first two, refer to particular species. So one of them is Portunus senius, okay, the guy shown on the right-hand side of the screen, the blue swimmer crab. I actually have a stuffed individual of this guy, okay? Part of it is broken, okay, because of the trip. But anyway, you can see that it's quite a large crab species, okay? In fact, in its native range, the species is native to the Western Indian Ocean. It is an edible species. It's regularly consumed in places like Bangladesh, India, Sri Lanka, and so on. In the Med, so far, there hasn't been, this, let's say, cultural shift to try to consume it, okay? So in places where it has reached large population densities, it's not consumed. Actually, people don't know yet what to do with it. There's plans, for example, in Tunisia, in the Gulf of Gabes, which is one of the hotspots, as we're going to see, where the species has established a foothold, to actually export it, because, it's become a big nuisance to fishermen, where it clogs nets as well, tears nets, fills boats, so even in terms of space on boats. Okay, so even if you Google, there's quite alarming images from Tunisia in terms of fishing boats, where literally the fishing boat, 3/4 of the fishing boat is taken up by this crab. So it is an issue, okay? Incidentally, along with the blue swimmer crab, this guy was collected in Malta about 4 years ago, okay? Yes, August 2015. That was the first instance when we started seeing a massive expansion of the species in local waters. So in 2015. This guy has been in the Med, was recorded in the Med since in Egypt, obviously, because Egypt is the closest to the Suez, in 1898. So this guy is one of the earliest, what we call Lesseps migrants, okay, one of the earliest tropical aliens to enter the Mediterranean from the east since the opening of the Suez Canal in 1869. So this is one of the first. But you might say, why 2015 in Malta? Its westward expansion has been quite slow, okay? Has been quite slow up till now. So in the last 3-4 years, we've seen a massive explosion of this species in the central Mediterranean. So in Tunisia, Gulf of Gabes, Malta, Sicily, okay? So that's why now all the renewed interest on the possible trophic impact of this guy, okay? I brought also another species with me, just as a side note, which is the pearl oyster, okay? Why? Because I said that this is one of the first Lessepsian migrants to make it into Mediterranean since the opening of the Suez Canal. This is actually the first Lessepsian migrant, okay? So if the Suez Canal was officially opened in 1869, this was recorded from the Med in 1870, so literally the year after. And this is the pearl oyster Pinctata radiata, which is nowadays very widespread in our waters. Okay, so that is the first target we have. Okay, second target, freshwater now. Okay, so leaving the marine, going on the freshwater, rather than focusing on a single species here, on a group of species, non-indigenous crayfish. And that is not an acronym that I invented, that I came up with, NIC, okay, non-indigenous crayfish, because as we're going to see in this presentation, there's a group of them, unfortunately. okay, on native freshwater biotic assemblages. And then within an Italia-Malta project, which is ongoing, within which we are involved along with the DFA and along with ERA, we also want to characterize the baseline, okay, for trophic feeding interactions within Posidonia oceanica seagrass meadows. So this work will hopefully happen later on this year throughout summer. Okay, so basically to characterize what's going on in terms of feeding relationship within this very important habitat. So let's start with the freshwater problem, with the freshwater system problem, invasion if you want. Okay, in fact here I've termed it NIC, invasion of Maltese freshwater systems. Contrary to what one might think, okay, and what is reported in the media sometimes, it isn't just a single species here. The guy on the extreme right perhaps is the most notorious one, okay, Procambarus clarkii. He is the red swamp crayfish, native of Florida, North America. But besides this guy, there are other species as well, okay, of non-indigenous crayfish. You have also Procambarus virginalis, okay, so staying within the same genus as the more famous clarkii, what is commonly known as the marmot crabs, okay, up there. And over down here, within this photo was taken within Chadwick Lakes with Leia, okay, with Leia Fiddin system. So actually large numbers collected in a single bait of the signal crayfish, okay, which is native as well of North America. Procambarus virginalis marmot crabs, I didn't actually specify the origin of the species because it's not completely understood, it's not completely known, what we know as a cryptogenic species actually. So the actual origin is not yet known, probably because of hybridization, okay, which is compounding things, okay, and some of these species actually might not be single species, but actually complexes. So the situation is quite complicated. It's not just these three species, there's also three others. So 6 species, all of which have been reported from Maltese freshwater systems. Not just reported, they are actually established. Okay, so narrow-clawed crayfish, now leaving North America, native of Central and Eastern Asia. Okay, Sharax, probably along with the Procambarus, the most destructive of them all. Okay, Sharax, Australian red claw, native of Australia, and then not a crayfish, but a shrimp, okay, bamboo shrimp, native, there's a typo there, native of Southeast Asia. Okay, so all 6 have been recorded in Maltese freshwater ecosystems, along with a couple of other species from Sicilian freshwater ecosystems. Okay, so this is basically a summary of where the species have been recorded. There is at least one other location besides this map, which is not shown here, which is Ain Rehana, which is basically Burmarat area, coming down from Bidnia to Burmarat area. So Procambarus clarkii are the yellow spots, okay, so you have anything from Tasarraflu, with L'Unziata, Ain il Papri. Some of these sites, for those of you who are familiar with them, some of these sites are part of natural freshwater ecosystems, but others are simply reservoirs. So they are not connected to, to the natural setting, to the natural ecosystem and so on, but just an isolated, even Ain il Papri, it's an isolated pond, basically, man-made, okay, man-made, like El Hofra, okay, Tasarraflu as well is isolated. So there the impact is somewhat contained, even in Nadur, another reservoir, but in other areas of the situation is much worse, because it's part of a interconnected system like with Leia, and not shown here. There's also El Bahriya. Okay, so unfortunately, why? Unfortunately, because like with El L'Unziata, it is one of the locations harboring the freshwater crab. Okay, Potamon fluviatile melitensis. So some considerations, okay, so far 6 different species belonging to 3 different families. originating in widely disjunct geographical regions. So we have originating from North America, Southeast Asia, and also Australia. So it's difficult to infer possible future ecological and trophic impacts. These are species which are popular in aquarium pet trade. Introduction for human consumption or as a fishing bait cannot be discounted. So, okay, probably the introduction was through the aquarium trade, but there might have been instances where these have been introduced for human consumption because they are actually consumed by humans. I am aware of at least one restaurant which offers these species, off the menu in an official way, or as a fishing bait. In fact, this photo, with the trap, those individuals were caught, by a person, by an individual who was interested in using these crayfish as a fishing bait. Deliberate release is very probable, given that the reservoirs are isolated from flowing freshwater systems, and obviously since there is a wide geographical distribution of records, involving both Malta and Gozo, and trade in at least 3 of these species, Progamborus clarki, Progamborus virginalis, and Astacus lenusculus. These 3 are included in the EU invasive alien species list for direct intervention. So any trade in these 3 species is obviously prohibited. Okay, and so the EU, as you might be aware, has this list, the invasive alien species list, IAS, which is targeting terrestrial and freshwater species. So far, there isn't a marine species list, and basically any species listed on this list cannot be traded, across the EU. And actually, these are shortlisted for direct intervention, for removal, for control, for extirpation, whatever. So at least 3 of them, in acknowledgement of their high invasive potential. Besides that, besides the potential ecological impact, there's also the ecotoxicological effect, because some of these species at least are vectors of either parasites, or they have been implicated in the transfer of toxins up the food web. So to higher trophic levels. The invasive potential of some of these species also is due to their habits. For example, Progamborus virginalis performs obligate parthenogenetic reproduction. So Sharax has been recorded from freshwater pools having a water temperature of 41 degrees. So I think it was in Slovenia. Again, not native range where it was introduced. Whilst most of these non-indigenous crayfish can survive prolonged periods of drought, and thus seasonality is not a problem. We have seen instances of biological control, even in Malta, where you have species feeding on them. For example, on juveniles, hedgehogs, feeding off the water on juvenile individuals, but this is offset by the high reproductive potential of these species, which obviously offsets, compensates for any losses, for any mortality through this predation. At the same time, obviously, they are highly invasive, and they're highly plastic adaptable. And there are concerns, obviously, as I mentioned earlier, in those areas where there are species of high conservation importance, with the Lunciata and Gozo, Schlendi, and obviously also Bahriya in Malta, and species not just the freshwater crab, but also our only indigenous amphibian, the painted frog. So, and we've observed in the field interactions in Bahriya with juveniles, with juveniles of potamon. Not with the adults, because the adults are large enough to fend off the crayfish, but the juveniles are not. And obviously there are other considerations like egg masses, laid by potamon, which can be fed upon by the crayfish, and so on. These crayfish species are highly, they are also scavengers, and they're opportunistic in terms of, not generalists in terms of of feeding and in terms of food sources. This is one publication through which the impact of Progamborus clarki, the red swamp crayfish, on indigenous amphibians on the Iberian Peninsula, so Spain and Portugal, was assessed. So in fact, the last sentence within the abstract, is particularly alarming when you consider that the ongoing expansion of P. clarki may eventually lead to a growing isolation of amphibian populations and ultimately to local extinctions and the permanent alteration of the amphibian communities in southwest Iberian Peninsula. So the ecological impact of these species is well documented. This is just a publication from 2006. Since then, there have been numerous publications from all over Europe and across their wider introduced range. For example, you find even virginalis in Madagascar nowadays. Now, on the blue swimmer crab, okay, this is the current distribution of the blue swimmer crab within the Mediterranean, so outside the native range. Okay, so it's up till a few years ago, it was basically limited to the Levantine basin of the Mediterranean, so the far east of the Mediterranean. Okay, so the Egyptian coast, the Israeli coast, the south Turkish coast, and Cyprus. But in the last 5 years, there's been a great expansion of this decapod, basically within the central Mediterranean. Okay, it is as I said, it is considered a Lessepian species, one of the first Lessepians known from the basin, slow westward spread, and it is an important food resource in its native range, but to date, not yet in the Mediterranean. Okay? Species has high invasive potential, reaching very high population densities. In fact, in the last 2 years, we've received a lot of reports from the south, mainly along the southeast coastline of the island of Malta. So it seems to have established itself very well in the Marsa-Shlock Bay area. Okay, the largest populations are known from there. But then there are probably smaller populations all along the eastern coast up to St. Paul's Islands. Okay, so quite a stretch, quite a stretch. And this is consonant with its preferred habitat. So its preferred habitat are shallow, muddy, benthic areas. Okay, so basically it invades shallow embayments. Alet Marco springs to mind, okay, in Bahar Chaq, for example, the Marsa Scala, harbor area, for example, and the best place in terms of spatial extent is Marsa-Shlock Bay, obviously, up to a depth of 50 meters. Females prefer shallower waters than males, mainly for spawning purposes in spring, okay, and the adult crabs are most active in summer and early autumn. Now, unfortunately, this species reaches sexual maturity in just 1 year. Okay, so the top photo was taken at Marsa-Shlock, showing, these were blue swimmer crabs for sale because they're being sold in Marsa-Shlock Harbor, along with a number of other aliens. So it's not the first alien to be sold at Marsa-Shlock. Okay. Rabbitfish, for those of you who are familiar with fish and aliens, rabbitfish Siganus ausalla maltese has been sold for quite some time. Even the blue cornet fish, as well, Fistularia. But now this guy is being sold for, I think, for the past 2 years, perhaps 3 at most. Okay, but this was from a single vendor. Okay, so 5 or 6 crabs, and this is a female carrying an egg mass underneath her abdomen. Okay, the difference between the two, between males and females, is quite evident. So as you will see from the claws, from the chelae, okay, in terms of size and so on. And so there isn't any published data, or very little, about its impact on Mediterranean benthic fauna. So all the existing studies on the blue swimmer crab refer to its native range, which is Persian Gulf and Western Indian Ocean. So there's quite a lot of studies from Iran, for example, and this is one of them. And I took this graph study, feeding habits of blue swimmer crab in the northern coastal waters of Iran, and you'll see that its food is mainly consists of crustaceans, mollusks, fish, and even debris. So it's quite an opportunistic, generalistic feeder. Okay, according to DAISY, which is the Alien Species Network operated by the EU Commission, the blue swimmer crab is competing for food with native biota, but no displacement or substantial impact on population level has been reported so far. Okay? However, it may pose obviously a risk of structural and functional change in benthic communities. Crab is carnivorous and a voracious predator, and its preferred food prey, as I said, already are crustaceans. Okay? And similarly to what we've seen for the non-indigenous crayfish, okay, it can host a number of parasites, okay, and itself it could serve as a vector for the introduction of other non-indigenous species in the Mediterranean. Okay, so again, to compound matters even worse. So as you see, the two species or the two groups of species that we chose, that we prioritized for SIA applications, you know, we chose them along these lines, that they are basically the ones which are most likely to exert a significant ecological impact. Okay, so these are some snapshots from preliminary work we did with the species already here at Aquabiotech. You'll see that we have four individuals here, 2 males at the bottom and 2 females at the top. You immediately see the differences, the morphological differences in terms of the chellae, but also in the width of the carapace, okay, which is wider, more pronounced in females, obviously to carry the egg mass. Okay, so as Alexia will tell you, okay, later on, and Brian tomorrow for those of you who will be here, when you're applying SIAs, obviously you don't just apply them on the target species if you're assessing the trophic impact, but also on the potential prey species of that particular species. Okay, so one thing we've started looking at is you know, the potential prey that this guy actually has in Maltese waters, because it's useless reading about the potential impact in Iran. You know, we have to see actually what this species is eating in Maltese waters. One of the problems we encountered is that this species, you know, because of the way it feeds, because of those massive chellae, tends to triturate and break down into fine pieces all of its food. So it's very difficult to identify when you empty the stomach under the microscope what the actual composition of its prey is. Okay, so you end up with a soup which is, you know, barely identifiable, and therefore you have to resort to genetic tools to identify the prey. Okay. So I'll stop there for now. Okay, I don't know if there's any questions, not on SIA in general, because obviously Alexia will speak about this, but about these 2 species perhaps, you know, or any monitoring that is going on for these 2 species. Yes.
You mentioned that the alien crayfish tends to prey on juvenile
Potamon, yes, freshwater crab, yes.
Since it's got this high conservation status, would you be performing any stabilized analysis on that species, or how will you tackle it?
We chose as a first let's call it pilot study, not to work at Bahriya, but to work in areas where there isn't the freshwater crab, you know, because it's still, you know, preliminary study, and basically we didn't want to, because some of the sampling can be pretty disruptive, because if you're sampling for potential prey, you have to go in the water, you have to do sweeps and so on. You have to disturb the seabed, you know, so it's better to work in an area which is, you know, enjoys a lower level of protection, okay, initially. So, and then, you know, we're not discounting that in a future we will work in areas where there is the, remember that with Elea Fiddin, even though there isn't the freshwater crab, there's the frog, at least, you know, so any impacts that with Elea are still, you know, of concern, you know, it's not, you know. But at Bahriya, we saw this through observations, through observations, you know. So and
There are other options with regards to prey species which can be
yes, and the species is highly opportunistic, so it will basically eat anything which it manages to ambush, because essentially both species are, you know, deploy the ambush. The blue swimmer crab, which stays burrowed in the sediment with its stalked eyes just popping out of the sediment, you know, and it ambushes with its claws, and the crayfish as well, you know. And once they catch a prey, they simply retreat. They swim backwards, you know, by the beating of their telsin. They retreat backwards, you know. So yes. And what was particularly interesting was the fact that hedgehogs, you know, could be seen picking out the juveniles from the water, you know, and even rats, you know. But I didn't mention initially the rats because, you know. So there is a broad literature. There are reports of eels, of anguilla, of eels feeding actively on the crayfish, you know. So yes.
Yes, you also mentioned a few alien species that we're using for human consumption.
Mhm.
Is this a negative thing? Will it be beneficial?
No, it's not a negative thing as long as it doesn't encourage the trade in live specimens, you know, or even in our individuals, our representatives are here, but even in dead specimens, I would think it's not that advisable, because to have dead specimens, you need to have some sort of farm or something somewhere, and that might lead to escapes into the wild. So in itself, it's not a bad thing, because if we look, for example, at the lionfish, one way it's being controlled is by encouraging people to actively fish it, cook it, consume it, and so on, you know. But one has to be careful that this does not encourage a trade in the species, you know, because then you start seeing commercial outlets sprouting up, you know, to fill that need, you know. So one has to tread carefully, you know. So even if you encourage people to go and catch the species, even that, you know, I'm very cautious about that, because you might catch then species which are, you know, of conservation importance, because the method you might be using might be indiscriminate and therefore disruptive, you know, and invasive in itself. So one has to be very careful, you know. My personal opinion is that the species, it's virtually impossible to control these species, especially the non-indigenous crayfish, you know, because the non-indigenous crayfish has very few environmental controls. The blue swimmer crab, you might get a particularly cold winter, particularly cold spell, as happens with the other aliens we're getting in our waters, where most of that species disappears. We've seen alien species which have declined over the years. For example, aliens which were established in the Grand Harbor, especially in the Menai area, in the Marsa area, have suffered a lot because of the closure of the Marsa power station. Because the Marsa power station had a thermal discharge into there, so the water temperature in the Menai was 5, 6, 7 degrees higher than the surroundings, you know. So and this is not documented in the media, obviously, but amongst the environmental benefits of closing the Marsa power station, there is also the positive impact that some of the aliens have declined in the Grand Harbor, you know. Okay. Any other questions? Yes.
What about Perchnong gibbesi?
Perchnong gibbesi. Perchnong gibbesi, Lightfoot. Okay, Lightfoot. It's an Atlantic species, so we don't consider it as such as an alien. We consider it as a range expanding species, but it's all over the place, all over the Mediterranean. So they found it even, I think, in Lebanon and Israel. So even though it's an Atlantic species, it's spread throughout the entire length and breadth of the Mediterranean. But over there we have more data, because as far as I know, there have been studies, even within the department, for example, of interactions with the native indigenous crabs. Okay, so with the Pachygrapsus, for example, I recall a thesis where they've released Perchnong gibbesi in the same aquarium with Pachygrapsus and seeing the interactions and so on. So but in terms of trophic impacts, yes, even there we might have problems, but I'm not aware of them. It's a good point to keep in mind because the numbers are massive of perchnon, massive. So over there the impacts probably are on the algae because it's a grazer.
But I think it's also a scavenger.
Okay. Okay.
So I've seen it coming up in large numbers in the evening, especially.
Okay. Okay.
And you can see it if there's material to be scavenged.
It is. And the problem with the perchnon is that it is not sufficiently large, probably, to be consumed. So if so it's not. It doesn't catch the interest of human consumers. That's the problem, So thank you. So
Would you mind to just leave your laptop there?
yes,
so I
for
can
sure.
actually
Yes, for sure. Let me close.
Yes.
Let me close.
That's great.
Okay. So up to you.
Thanks. Okay, so again, thank you for coming here despite all the changes in time and for making it today. And we are going to start now to have a look more in depth on what stable isotopes are and what they are actually used for or can be actually useful for. So before that, I just, as we mentioned today, the lecture was supposed to be given by Dr. Brian Hayden, and he's in transit. Hopefully he's going to be with us in the afternoon. Just to give you this lecture, so basically we are kind of sharing. Brian is with us, let's say, into the lecture with the material that he provided, so we can kind of combine a couple of different lectures together to make it work for today. So in terms of his main research activities and type of projects that he carried out in the past, and doesn't work on the screen? Wonderful. And is mostly working on climate change, productivity, biological invasion. But this is actually just very, very broad thematic themes on which he worked. They mostly actually are related to conservation in the past. And currently is the science director of the SIN Lab in Canada. And the SIN Lab is a laboratory that is dedicated to the analysis of stable isotopes and is mostly working with carbon, nitrogen, but also hydrogen and oxygen. And now we are going to see how these different isotopes are going to be useful for us in research. And is basically running a semi-commercial lab, has a very good staff of technicians and healthy population of PhD students and postdocs. And through the various projects and collaborations that he has basically been working on 5 different continents. And now also we are starting collaboration here in Malta. In terms of similarities of career path, that's actually where we met, is that myself as well, I can describe my life as a life aquatic in a sense. I started working with stable isotopes back in 2005 when I was at the University of Dublin carrying out my PhD, and that's where I met Brian. Back then I worked on freshwater species and trophic interaction of brown trout and an invasive species that is roach, Rutilus rutilus. Brown trout is a species that is extremely important, especially for anglers. There is a lot of, let's say, energy and resources put in try to protect streams and lakes where brown trout spawns and lives. And so we were trying to figure it out if this invasive species, roach, that comes from Eastern Europe, was using the same trophic resources of brown trout. And maybe to figure out why the brown trout stocks were collapsing. It turns out that actually was mostly due to eutrophication and due to agricultural runoff in lakes and streams. I also worked on using not only stable isotopes and continent analysis for this type of work, but I also use molecular tools that helped having shed light in which is the population structure of brown trout in natural condition. Then I moved back to my first love, that is marine, and worked on ocean acidification. So again, try to tackle the effect of climate change on a broader spectrum of trophic webs in marine environment, and also started collaboration and go back working with Brian on flamingos. So, okay, it doesn't swim, but still is connected with the aquatic life because it's a bird that actually nests in areas that are brackish or very close to freshwater resources. So its food web that belongs to is actually an aquatic food web. So that's the way we met. Currently, at the moment, I'm a senior scientific consultant at Aquabiotech here in Malta, and we are using stable isotopes for a European project like the TAPAS project. The TAPAS project, we are going to have a look at it later on because it's actually a nice example of how to apply stable isotopes for research, but also for having a very applied research in terms of results, because we are using it for exploring the effectiveness of integrated multi-trophic aquaculture system, and also working on other European projects like BETOS. Now, we are all here because we want to know a little bit more about stable isotopes. What are they, how to use them, for how they can be actually useful. And as Carl Sagan reminds us, after all, we are actually made of star stuff, in the sense that in one way or another, the atoms that are part of our cells and our live matter are coming from the stars, ultimately. Now, if we look at the distribution and the composition of main chemical components in our bodies, we are mostly made of oxygen, carbon, hydrogen, nitrogen, and then in smaller percentage we are made of other things. But if we report that and we have a look at the table of chemical elements, basically all these ingredients, all these elements that make up us are actually in nature not only one form, but multiple forms. That's where isotopes comes into play. So for oxygen, carbon, hydrogen, and nitrogen, that actually are the most common used isotopes for research, there are multiple forms in nature. If we look more specifically, if this is hydrogen, the most common and abundant form in nature, then there is a nitrogen with an extra neutron in its nucleus, that is deuterium. Now, because the difference between these two isotopes or similar forms, the difference is just one extra neutron. The extra neutron in the nucleus does not change the chemical behavior, the chemical characteristics, or the physical characteristics of the hydrogen itself. It just makes it heavier. That's it. That's the only difference. So if we have a look instead of carbon, again, very, very common in organic matter, we have carbon-12, the most abundant in nature, so we're talking about 99% of it, but also a form that is carbon-13 and carbon-14. Carbon-13 compared to carbon-12 is different just because it has an extra neutron. Carbon, and is stable, does not decay in geological times. That's why we are starting to talk about stable isotopes. Then there is another form that's carbon-14 and is radioactive. So this means that carbon-14 does decay in geological time. So it's something that is very commonly used also for various diagnostics or research application. But what we are going to look today is only about stable isotopes. So isotopes, forms of similar forms to the one that we know already, but they are a little bit heavier, do not decay. And if we look at this modified version of the periodic table of elements, the one in pink are the element in pink are the one that have multiple stable isotopes in nature. So, for example, nitrogen or sulfur have different type of isotopes. There is not just nitrogen-14 or carbon-13. There are others, but there are very, very, very small amount in nature. So we try to work only with one that is most abundant, if you like. Pick up in terms of what is an isotope. A chemical form that has just an extra neutron. It does not decay in geological time, and vast majority of research is actually focusing on carbon, nitrogen, oxygen, hydrogen, and sulfur, because this is the most common elements present in the organic matter, and that's what makes us up us and all the other species that live around us. So that's why we focus on this. And it's nicely represented here in the logo of Ecological, that is the most famous international conference for the application of stable isotopes. As you can see here in this little drawing, in this little vignette, we have carbon 13, hydrogen, well, deuterium, nitrogen 15, oxygen 18, and sulfur 34. So it's important to keep in mind that there may be other forms as well. The important thing is that once we, when it comes to measurements, once we want to measure the abundance or the value of the heavy form versus the light form, what we actually measure them against the standard. The standard are different type of material. It could be, it's actually for carbon PD belemid, that sounds kind of mysterious, and is actually a piece of, is the carbon present inside a piece of stone coming from an asteroid. Don't ask me why, but that's what has been chosen for standard. For nitrogen, instead, we use air. Exactly the air that we are breathing here is the standard that we use for measuring nitrogen, against which then we can discern which is the ratio of the heavy versus the light form. In some cases, because of the mathematics behind it, that I'm not going to go into details today, for carbon, the values are negative. And also the notation, because the amount in terms of ratio of the heavy versus the light form is very, very, very small, the notation is part per thousand or per mil. That's one thing. We are going to have a look at some of the results coming from isotopic research. You're going to probably hear me saying carbon part per mil, and that's the notation both for all the isotopes, because the amounts are very, very small. So one of the first rules of isotopes, when we are going to look at that in the environment, is that every single of the isotopes we've been talking so far is distributed unevenly in the environment, just because of natural conditions of the environment we are going to look at. So this is due more specifically to the different biogeochemical cycles that happens in nature. So if we look, for example, at nitrogen, we can see that 99% of nitrogen on Earth is actually present as molecular nitrogen in the atmosphere, and then in the ocean as dissolved nitrogen. Very, very small amount then combines with other elements, and then we can have different form of oxidations and nitrate, nitrite, ammonia, and so on and so forth. And it's really, really important to keep this in mind, because nitrogen can be only available and in certain forms, and the availability depends on the biogeochemical cycles, but also biological activity. So depending on the type of the environment we are going to study, the availability of nitrogen, like all the other isotopes, can change just because of this interaction. In terms of nitrogen cycle, the availability, I was just saying that the nitrogen controls controls the production in many systems, because nitrogen, like sometimes phosphorus, can be the two main limiting factors, for example, on the productivity in oceans for phytoplankton. And to understand it, let's say this distribution, it needs to consider which are the biological cycles or nitrogen, and they are quite complex also, because now shedding lights in what the microorganism activity are contributing into the, making nitrogen available for pick up by other organisms. If we want to look at the, let's say, focus on the aquatic component or the aquatic habitat, we have that nitrogen coming, let's say, in the atmosphere, goes through the terrestrial system, then gets released into the aquatic one, and then in the various forms of component of nitrate, ammonia, and so on and so forth, can be made available through the process, through a biological process, through bacteria. And if we look in a more like wide scale, looking at basically space and time, the availability of nitrogen can be measured, and you can create maps like this one, where there is a latitudinal variation of delta 15N. The delta 15N is the notation of the ratio of nitrogen of light versus heavy form, and this is reflected by difference in biogeochemistry, again, because of the biogeochemical cycles I was talking about as before, but also it depends on the activity of the organism within the water column. In this map here, we are obviously covering a very, very big area. This is basically all the Atlantic Ocean here. And the black points shows you where the sampling has been done. We're talking about only surface water. Doesn't matter though, but the point is that you can create maps like this that helps you, let's say, foresee or give an estimate of what would be the nitrogen delta 15N present in the specific environment, and that's actually very, very useful, and then we are going to see why. In terms of carbon, again, carbon can occur in a variety of compounds on Earth, but for our purpose, what is really important is organic carbon. Another form of carbon is the inorganic carbon, and sometimes can come in our samples and confound a little bit what we are looking at. And in terms of cycle, again, it's a quite complex cycle because it basically links both the terrestrial with the aquatic one. The CO2 present in the atmosphere gets diffused into the aquatic. There is a lot of animals and basically plant activities here, biogeochemical activities and processes, and then it connects as well with the terrestrial element. So again, the availability of carbon in the form of organic carbon, that's what we want to trace into the food web, could be depending on the different activities and the processes that are happening in the environment. Again, in terms of cycles, without going into the specifics of how much carbon is produced in different type of environment, let's say that the most important thing is taking into account that there are two forms of carbon, organic and inorganic, because this is going to be recurring especially during the practical later on, because we have to try to keep away the inorganic carbon. And if we cannot, when we sample live tissues, we need to go through a process of acidification, because it can confound our results. Regarding carbon, the interesting part, the interesting information that we can get through carbon is that it can help us discern which are the main sources of energy for a given organism. In the sense that each compartment into this little drawing here, basically, each compartment and each habitat represents a different habitat. Each habitat has a different base value of carbon, as you can see here. As we were saying at the beginning of the lecture, carbon is the notation of carbon is negative and is in part per mil. So marine plankton, we're talking about averages, has an average of minus 21 per mil, seagrass minus 10 per mil, and depending on the metabolic pathway of which the plants are making photosynthesis, C4 plants have a distinctive minus 12 per mil carbon signature, and C3 plants have a very distinctive signature of minus 27 per mil. So if matter coming from the degradation or the debris of these plants from terrestrial gets into the aquatic environment, based on the different signature, as we said, very distinctive on carbon, and being picked up by some organism, I can actually track where the resource of their food was coming from, based on the fact that I know which are the average of the carbon signature coming from this different compartment and so on. For example, if into the marine system I want to study a species that is a detrivore and is picking up some detrivores close to a river source, I know that the matter coming from river, the POM or DOM, is actually a very distinctive signature. So when it gets incorporated into the organism, then I can actually read that, because I have an idea of how to have a sense of the landscape. So this on a more broad spatial scale, what does it mean? That actually I can do what I did for nitrogen, and sample across all the Atlantic for delta 13C ratio and have a map. This is called carbon and nitrogen isoscapes. And you can repeat that for any other isotopes you want, provided that you have enough funding and a good partnership in this kind of sampling, because it's quite intensive. But those sampling point, even though they look a little bit scattered. Through modeling and through some estimation, it's possible to actually have an estimate of what's happening in between. So you really create a map that helps you establish the baseline signatures of your carbon or nitrogen, as we saw a few slides before, and help you then try to read the environment for kind of very, for the application that can be very, very useful. So far we talked about carbon and nitrogen because are the most commonly used, especially in trophic ecology or ecology in general, but very useful as well are the isotopes of hydrogen and oxygen, because they are important elements present in water. All organism needs to live water. So similarly as I did for carbon and nitrogen, I can actually map water into the system, in terrestrial system, and have a map based on basically tap water. I don't know if you can actually see, it's actually very small here, but this map of the US is basically done using tap water samples. Again, the samples are the black dots here, and this gives me a baseline for delta O18. And then I can use that for example, tracking migration patterns of birds, or if you want to see where is a food source that you find, I don't know, in Milwaukee, you buy in Milwaukee, maybe it wants to know where the food has been produced. Based on this map and analyzing delta 18O in the food, in the sample that you want to assess or assign, you can track it back. So again, it's a tracking device in a sense, in its own accord. And the same is for deuterium. Okay, so again, a very, very powerful type of application. And since, again, what I want to stress is that despite few points here of sampling, then you can, through modeling and simulation, establish prediction maps. So there is certain amount of uncertainty, but then the more samples you have, as usual, the better your map becomes. So the second rule about isotope ecology that actually is very interesting is that in a way those isotopes are conserved in the organism that picks that up through feeding, or through, if you are a plant, through basically incorporating the basic nutrients within the food web itself. So all our consumers in a way assimilates the isotope through the food, as we said, and then retains that in their own body. And this has a very, very important implication in trophic ecology and in other applications as well, because we can say that in a way, because of the various availability of isotopes through biogeochemical cycles, and we have created some maps, as I showed you, about the distribution through global, regional, and site-specific. And let's say, it actually relies on the isotopic difference that we can actually pick up depends also on the type of organism we are going to go study. But ultimately, what we can get in terms of ecological information, that's why we are here to see the application to aquatic ecology, is where the consumers are eating, what they are eating, how much, when they are eating, and how they basically change the trophic position into the food web. So we are not going to talk now about the isotrophic niche, because that's something that we can have a look at if you are going to be available tomorrow or after we go through the practical, because it's something a little bit more complex. But ultimately, when we put all the information together for carbon and nitrogen, we go out there and samples and look at the results, is something like that. That's basically a biplot in which on the x-axis we have delta 13C, so basically the isotope ratio carbon. On the y-axis, delta 15N, that is the isotope ratio of nitrogen. Carbon, as explained before, helps you to discriminate where does it come from, the main source of energy in terms of carbon for an organism, whereas delta 15N helps you discriminate where your organism or your, let's say, your target organism is located within the food web. So allows me to tell where is this food, the trophic level it is occupying at a certain given moment. Now, what I wanted to mention, looking at this plot, that is one of the first one that was probably done hand-drawn and then published on the paper in 1984, is that for carbon and nitrogen, in nitrogen there is a constant increment of the amount of nitrogen from prey to predator. And that's constant because we said basically we assimilate the isotopes from the food we eat, allows us to assess that from one trophic level to another there is a difference of 3.4 per mil of nitrogen. So this consistency and constant allows us to, having sampled our target species and having the baseline that either come from the map that I showed you before, or just going in the field, as Alan said before, and collecting physically the baseline, I can actually tell my crab or crayfish where does it feed within a certain food web. So this is a practical application of stable isotopes. Again, also from a lot of literature, lots of papers, lots of work that's been done in the past, we managed to assess that an average value of increment for delta 15N is 3.5 per mil, the one I was just talking about, whereas for carbon is 1 per mil between prey and predator. So knowing this gives us a good estimate. But if you know exactly what your species is characteristics increment is, even better. It could either come from literature, or if you really have the chance to sit in a lab and do your experiment, you can do a feeding experiment on your own, because this that I call so far increment is called fractionation. Now, the fractionation is exactly the process by which an organism eats something through metabolic process. It uses mostly of the light form of isotopes because of thermodynamics and kinetics. And then in the tissue are left only the heavy isotope. Then if let's say I'm the species that is an object, I'm eaten by someone else, then someone else is going to be enriched by a certain amount of in that heavy isotope. And that enrichment, that constant increment, is the fractionation. So fractionation is related ultimately to biological processes. And this goes for plant and goes for animals in the same way. Now, these are average values, but I invite you that if you're going to ever use stable isotopes, look up for previous papers or previous literature where people have been working with a species that is either your species or something very closely related. Because also one of the very important elements, this we're going to even talk more about it in the lab, is that we have to keep in mind the turnover of elements within an organism, because carbon, nitrogen, sulfur, oxygen, so on and so forth are not constant. What I eat today in my food that we are going to have right now is going to stay with me for a little while, but then my cells are going to go and turn over, and I need some new carbon and nitrogen coming from another food source. So this information has to be taken into account when you're going to plan your study, and we are going to talk about that a bit more in detail later on. So, putting everything together in terms of information from nitrogen, carbon, the food webs, what we have and what we really need to keep an eye on, as I said, are our baseline. Let's say that my study target species is this seal, could be Phoca vitulina or whatever other seal. But in order, if I want to sample some tissue material from these seals, like could be the whiskers or the fur or whatever, or if I found a few dead seals, I can get some muscle flesh. Anyway, whatever samples comes from these animals, in order to know where this species is placed within the food web, I need to have information from the baselines. If my target is a marine environment or a freshwater environment, the bigger the environment, like a big lake, body of water, or like in this case is a coastal zone, I need to have all the representatives of the various possible food web within that environment. So in this case, I suppose that there is a planktonic or like a planktonic kind of food web here, but also a benthic food web. So in both cases, I need to get baselines. And there are studies before that have been showing which baseline is more let's say, appropriate considering the target organism that you're looking at, in order to be representative of that specific part into the food web, especially if you don't have any information whatsoever about your species. Like, for example, the study that we are doing with Alan. We don't really know much about what the crayfish or the blue crab, since they are invasive, are going to be using as a baselines in Malta, because they are invasive. So we can just kind of guess from what's coming to the literature. We can have a good estimate, but to be sure, when we go out there sampling, we try to represent as much as possible the environment we are going to work. So if we're talking about the blue crab, I would actually, just rule of thumb, generally speaking, going for some bentonic organisms, but also would represent something that is in the water column, some part of the pelagic realm. So that just, I'm sure, and it doesn't hurt you to sample a little bit more than then going back to the lab and then realize that you didn't have enough. Because again, there is a temporal variation, not only a spatial variation in isotopes. And this can't, once you're back into the lab, you cannot repeat the samples, unless, because you cannot go back in time. So, again, as I said from before, you can try to get good estimates relating, if you don't have the chance, maybe you get back into the lab, oh God, I didn't get enough baseline, what do I do, what do I do? You can try to get estimates related to the baselines from papers or from other work of other people, and try to have some general averages. As shown here, each baseline, again, has a very unique signature in terms of carbon and nitrogen, so that it helps you discriminate and better place into that, if we look again back here, where where seal was eating. So if it's more towards this part of the graph, it means that as a carbon source, it was more pelagic. If it's gonna sit your value more in this part of the graph, means that the carbon that it is using as a resource is more coming from a benthic food web. So this helps you basically put together the various information coming from stable isotopes. And I don't know if you so far have any questions, because in the next few couple of slides, what I want to talk to you about is further practical application of stable isotopes, a real project where we are working together in collaboration here. Any question, doubt, uncertainty?
Well, we are talking about stable isotopes, aren't we?
Yes, we are.
Okay. No, because clearly, if there's a pattern in the ratios of stable isotopes, there might be another ratio indicated also in the unstable isotopes. And the unstable isotopes are easier to meter.
Which are the unstable isotopes? The one that decay?
Whatever the element you're looking at. I don't know, it's just an idea.
Okay.
But we are looking at the stable ones.
Yes.
Okay.
Yeah, the radioactive one.
Yeah, because I mean, in this case, you would use what? A mass spectrograph, no?
We use an IRMS, that basically is isotope ratio mass spectrometer.
Okay, mass spectrometer. Because it's certainly easier for the unstable isotopes. And you could have instances where you could have fingerprints of unstable isotopes in one particular chain. Maybe a food or living matter, whatever.
Yeah, but
potassium, carbon, everything else, and that's possibly even easier to obtain. But just an idea, which I think once we had attempted to do something with food as regards feeding fish farms and the way the fecal matter settles on the bottom and such that. And,
but because they are radioactive, as you say, they are or unstable, as you're saying, I cannot use them as a tracker or biomarkers throughout the whole food web because they change.
well,
So
yeah, exactly because of that, you could even have, in some instances, some details which could pertain to that situation, and they could give you certain details. But that's maybe a different idea, different.
There are application of radioactive isotopes. There are lots of. There are lots of, but not for this type of issue.
Medical.
Yeah, for medical. Yes. for archaeology. You can use them as basically capsule time machines. Yes, but the stable isotopes, they work differently because the applications we are using them for are different. Another question?
Yes. So basically, we're always measuring the ratio between stable isotopes and normal isotopes, right?
Yes.
Looking at the ratios.
Is a ratio between the heavy and the light form.
Yes, that's understood. And you said there's seasonal variations, which is
Yes.
also clear to me. What range do these variations have? What range are we looking at? And the second question is, you said you can take samples, tissue samples, but within the body, are these stable isotopes distributed equally among the cells? Because from a whisker to a flesh or an organ sample, I think there must be a difference, no?
Yes, there is.
So that was my two questions.
Okay. So the first question, yes, stable isotopes do vary in space and time. If we go back to one sec. One of these maps, it could be either for carbon or nitrogen, doesn't really matter. But these are averages across, big landscape, so in terms of space, they do change. Colors are associated to delta 13C, that is measured in phytoplankton. PLK, that's what it means for, okay? So as you can see, there is physically a geographical variation due to biogeochemical processes, availability, and so on and so forth. Time, yes, it does change with time, because, for example, in this case, since it's associated with plankton does change
No, I
very.
understand the change in time. I just want to know what the range of changes. Like this map would look the same. And
Well, here we have
the differences between the area would stay stable, I would assume, no?
within one small area on local scale, it stays stable. And we're going to talk about that later on in the next few slides when we are looking at real cases, like study case. But yes, but if you can see on top of the scale, it goes from -30 to -15, so there's a lot of variation there. But on a small scale, you get much more consistency. But in time, if you, let's say, you have a sample, I don't know, a study that has a time of 1 year. Every time you go out, if you're representing seasonality, you have to go there and sample both your target and your baselines because it changes, especially if it's an aquatic environment. There are environments that are a little bit more stable for the way that this biogeochemical process happens. But in aquatic, I would go there for safe measure and have measurements.
The change is like 1 promille or 2, or is that
Could be even more.
unpredictable? Or?
Yeah, I wouldn't be able to predict because unless I have a map like that, and I have information from seasonality. But if I'm gonna go blind in a new environment, new research project, I don't know. I just have to collect samples.
And
Yes.
the difference between tissue samples?
Between tissue samples, the interesting part, because of the turnover, every tissue sample gives you a different time window. As in, from the moment I eat to the moment I incorporate that in myself.
That would be take into consideration the calculation of the distribution within the body.
In the case we look at physiology of the organism. So you need to take into account the muscle. For example, if I'm talking about fish, just because I'm a fish person, if you talk about fish, muscle gives you a window time of what happened in that, for that animal in the last 3 months. If you look about blood, it probably is going to be a day or 2. If you look about liver, probably it's going to be a week. If you look about scales, it's going to be a lifetime. So depending on where you're going to sample on a scale, you're going to see where the fish the whereabouts of the fish. And the same goes for otolith. Depending on the growth, on, let's say, the turnover, the way the metabolic pathway or the specific tissue, then you have to take that into account. If it's an organism that is actually cold-blooded organism with a lower metabolic pathways, or if it's a species that lives in the tropics, all these elements have to be taken into account when you go out and sample. So, yes.
I also have 2 questions.
Yes.
The first one is, does this work, or are there challenges for like migratory species or species that have like a highly varied diet?
Yeah.
Like we can't do that. And then the second one is, has anyone tried to do this for like human population?
Yes. Yes.
Okay, cool.
Cool. I'll show you actually, connecting to your question, Brian, just to answer them. I'm going to show you the real case I wanted to talk about. These data are unpublished. It's Brian's work. So we were talking about yesterday while he was in transit. And it's actually a very, very exciting piece of work because he works with salmon salar. So Atlantic salmon stocks collapsed At one point, we had very little Atlantic salmon. As a type of species, is a migratory species. It does reproduce in fresh water, then there is a metamorphosis process, and then it goes swims up in the open ocean. And is a highly migratory species and has actually anadromous species, so it goes spawning back to the river when it was born. Now, the interesting pathways is that the animals that are born, I don't have a point, the animals that are born here in Ireland, UK, France and Spain go out and go eating basically around Greenland, Newfoundland, and so on and so forth. Now, as I said before, different tissue give different type of information. The scale that is being probably laid at one point in the lifetime of the fish can carry much longer information than it could be muscle or blood or liver, okay? So if we sample on the outer ring out here for stable isotopes, carbon and nitrogen, probably I think that's what Brian did, and then we look at the animal, the adult, but also we compare with the young, the juveniles that are still living in UK, Ireland, because before they can go out at sea, they have to go through a process called smoltification, and it actually happens around the first year of age. So before they do the metamorphosis, they stay for a little while in their native rivers. So if I have the juveniles and I compare with the adults, when I go out there in the mixed fishery in the Atlantic Ocean, and I pick one of the salmon and I compare the scale of the adult versus the scale of the juvenile, I can actually get basically stock assignment. Now, in these circles here, in these three plots, again, we have delta carbon 13C on the x-axis, delta 15N on the y-axis. Cook, hatchery, and SIS, three hatchery samples. UK and Ireland obviously is a wild population. And if you can see, these green dots are been some salmons that have been collected. I was actually talking to Brian yesterday in Greenland, and it seems they can actually get very close to this sample here collected in UK and Ireland. Other animals that have been found in Newfoundland are actually closer to this hatchery here. So we know that different animals that have been released either from an hatchery or coming from the wild are going eating in different feeding grounds. So this is another way of using stable isotopes with a very a technique that is actually very little invasive because it's a scale. I don't have to either kill or do biopsies or harm the fish. I can either also go, for example, I used to do it for trout, go into angling competition and get a little sample. So I didn't have to go really in the field or go into the lake and kill some more fish, but just go to angling competition, collect a little sample, and then do my analysis. So it can be also the sample size you need is way smaller than going out there and doing the usual sampling for fisheries through, I don't know, gill nets or counting the animals and so on and so forth. It can help you establish a better idea of what's happening in the fish stock and the population. The very last examples I want to talk to you about is a practical example. We changed species, is not salmon anymore, using stable isotopes to assess the feasibility of use of integrated multitrophic aquaculture systems in Malta. We are working in collaboration for the TAPAS project in the Marshalls Lock Bay with the MFF. What we want to see is that if using invertebrates like sea cucumbers, we are going to be able to help bioremediate the water column out of the effluents coming from the aquaculture activities. This is our experimental design. The setup has three locations, 0 meters just underneath the fish cage, 10 meters and 25 meters. We have 4 cages containing sea cucumbers, and we are going to test them using stable isotopes, but also looking at the biometrics to see if they're actually picking up the nutrients coming from the fish farms. And this can be done using nitrogen and carbon as basically markers or tracers, okay, into the food web. Now, the only results that we have so far are coming from what we call, let's say, an exploratory sampling. We didn't know anything about the bay. We didn't know any information, we didn't know what was going there, but before we could actually set up our nice experimental cages and everything else, we needed to know what were the baselines, which were the basic values of carbon and nitrogen present in the bay. So what we did, actually what they did, because it was before my time here at Aquabiotech, they went out there and they sampled the sample Posidonia, that is here. We have down here fish feed. Fish are the one up there, and then we are here sedimented organic matter, and then the organic matter both collected through sediment traps, so representing what's in the water column, and also the bottom of the sea. And at least we have some elements, some information that we're going to use later on once we're going to sample and process our sea cucumbers. So that's another way, that's a real practical application way in which you don't know your environment, you don't know your system, you go out there and you need to start to get information. But as a general rule of thumb, just really know your system before you go out and sampling, because it's better not to have any sample, it doesn't mean anything, rather than having tons of samples and you don't know what to do with them. So these are the little ones that are now in the cage, the Uluturia poli, and soon enough, hopefully, we are going to have more information and more results. So this is just a grazie from Brian's. Again, hopefully he's going to be with us soon. And that's from me for today. And if you have any further question, yep.
Yes, I'm just curious whether this technique has been used to assess the status of the ecosystem per se. So I'm asking for one of the descriptors of the MSFD is to assess the trophic system as a whole in the marine environment. And I'm questioning whether such technique was actually used to assess the ecosystem as a whole and not just target species.
Assess it as a whole, as in the productivity, if it's polluted, if it's...
The status in general.
The status in general. You can do the...
we are speaking more on a specific
target species. Yes. There's been done for, basically what we did when we went out in Marshall's Lock and we didn't know what we were facing or what we were doing. That's basically what we did. It was just a descriptive form. Then if you are interested in see how carbon and nitrogen are moving through the food web, that's another way you can do it. But yes, there are descriptors and also there are average values that they can try to figure it out if it actually is a healthy ecosystem or not, yes.
Okay.
Yes.
Alexia, we've already mentioned confounding factors like migration, for instance.
Yes.
In the case of aquaculture and the food that actually ends up uneaten and goes to the bottom, the original source of the food could be different. Mm-hmm. So that could be another confounding factor.
But that's why we are using stable isotopes and we collect baselines, so that once we put all the elements on that plot, we should be able to tell if the sea cucumbers is actually eating stuff coming from the fish farm or not, because together with the baseline around the sea cucumbers, we are going to collect as well, we already have actually points on this graph about the baselines related to the fish feed, the fish feces, but also the surrounding environment. So we are going to have Posidonia, we are going to have the sediment at the bottom of the cage, we are going to have the sediment in the water column. So then reading what's in the muscle of the Uluturia, I should be able to tell if the carbon or nitrogen coming from assimilated by the animal is originated from the fish feed or is more originated from Posidonia fibers. I don't know, just making an example.
Okay, I can understand that, but if the sources of fish feed are different
with, as a soy, well, we have the ingredients. Yeah, we have the ingredients of the fish feed.
Okay. So that has to be tested as well.
Yes.
Has to be.
Yeah.
And if it is frozen fish that is being, for instance, given to tuna, if it's
okay.
Tuna.
In the case of the tuna, yes.
Right? So
we need to get to sample that. In the case, we would need to sample that as well. Yes.
Yeah. The other question is the applications for terrestrial trophic, because I'm sure that.
Oh yes, they can be applied to any ecosystem. Yeah.
But are you dealing with that as well, or
personally, no. Personally, at the moment, no. And in the past, when we were working in a freshwater environment, we would look at the input of terrestrial from rivers in then the food web of the trout. So that would have been my focus. But yes, there is a lot of work done in terrestrial. A typical example is also using hydrogen and oxygen as isotopes, but also you have carbon and nitrogen, and there is a lot going on with microorganism breakdown of the various components. There is lots of studies on that. In a way, also the flamingo study I was talking about kind of links the various habitats.
I'm sure that with the composers, for instance, it may be very interesting.
Yes,
to follow them.
yes, yeah. Also because nowadays, I didn't have the time to show you, but nowadays the papers that are coming out are never only using solely stable isotopes. There is always genetics with it. So in terms of microorganisms, the thing that can help us shed light is next generation sequencing on a bunch of soil and see who's there, who's doing what. The problem, not the problem, but the thing is that we are opening a big Pandora box. The more information we get, the more we realize that it's very, very complex. The biogeochemical processes are very, very complex. It's not so easy and trivial as going through a graph. It's actually much more into that. And yes, the values that we get are estimates. Just let's keep that in mind. They are not absolute values themselves. So
meta-analysis of
yes,
usually.
exactly. To say the truth, that's the reality. We can have a sense and an estimate of what's going on and describe how energy flows through different compartments in the environment, but ultimately we're just putting different dots on a paper.
Scratching.
Yes.
Thanks.
Yeah.
Thank you.
Why there is a big difference between Posidonia and fishes, for example, in this graph?
Why there is because these fish are coming from the fish farm.
Okay.
So these fish, they never ate Posidonia in their life, allegedly. This one comes from the environment, the surrounding environment. So basically Posidonia, the difference within the Posidonia, let's say the blue and the green line, that's what you mean, or from the fish?
They are.
Okay, because Posidonia is a photosynthetic organism. The different lines and the colors correspond to the different parts of the plant. We separated them in roots, rhizomes, and leaves. So each part or each compartment of the plant has a different thing, biological point of view. So in terms of metabolism is different. So that's why we tend to separate them. In terms of why fish and plants are so different is because the type of carbon and nitrogen they use is different, because fish has been fish fed, a fish feed. I think if I remember correctly, soy, fish meal, some other ingredients, and anyway, makes it higher up on the food web. So, yeah. I don't know if there is any further question, I don't know.
We can maybe do that.
Okay, later on. Okay.
So, I'm taking out the yes. So for the tour downstairs, we'll be looking at some tanks, some fish, some grass systems, and everybody's coming down? Right, some basic instructions: don't feed the fish or the workers.